So hi everyone, I'm S.Jones from Brown University, and I'd like to thank the organizers for inviting me to participate in this very exciting workshop. I'm glad to have the opportunity to discuss some of the current challenges and opportunities in decoding the neural mechanisms and meaning of human EEG and MEG signals at the signal trial level. So I'd like to begin by giving you a sense of where my perspective on this topic comes from and briefly describe the challenge in human neuroscience that my lab aims to address. So we use MEG and EEG to study somatosensory perception. And we all know that these techniques are really powerful, but one of the downsides is that it's still hard to interpret what's going on at the cellular and circuit level. And this circuit level understanding is critical to know why these signals correlate with behavior and to target treatment when they disrupt. Now, invasive recording and modulation techniques in animals are ideal for dissecting circuit activity, but it's hard to do this in humans. And so to bridge this gap, my lab has been developing computational neuromodeling tools that act as the translator between what we're measuring at the macro scale and what's going on inside the head at the micro scale. And with funding from the Brain Initiative, we've recently turned this modeling framework into a user-friendly software tool for the community to begin to interpret the neural origin of their signals, and we're calling it Human Neocortical Neurosolver. And so in developing this integrated approach, we've come to understand some of the challenges and opportunity in trying to decode these human signals at the cellular and circuit level, and that's what I'm going to focus on in my talk. And so there are many new and exciting advances in applying complex analysis methods to decode how human signals relate to behavior. And here I'm highlighting some of the common types of data analysis that we apply to our data with increasing level of complexity. So with minimal level of complexity, we're looking at activity from a single brain area. And typically we focus on things like event-related potentials or low-frequency brain rhythms that emerge in the so-called alpha, beta, and gamma bands. From there, we often consider functional connectivity, where again, we're focusing on rhythmic activity typically and looking at the coordination of power or phase locking in the different frequency bands. And there's lots of exciting methods being developed these days to classify network states that regulate behavior with machine learning algorithms or graph theoretical methods. And again, these typically focus on things like power and phase locking as the underlying metric to classify. Now, with any of these methods, there's a major gap and challenge in answering the question, how do we connect these complex analysis methods to the underlying circuit level mechanisms that are regulating behavior? And a path forward that we see as viable is to first develop a better understanding of the neural mechanisms that are underlying the single area phenomena. And from there, we can use that as a building block to understanding these more complex decoding algorithms. And so next thing I'm going to give you an example of how we've been trying to develop a better understanding of what's going on in the single area phenomena using an example from our somatosensory perception experiments. So one of the major challenges we've come across is that averaging that's applied to improve signal to noise can often distort oscillatory features. And this creates difficulty in decoding their meaning in more naturalistic behaviors. And so to highlight this issue, here I'm showing you data from one of our tactile detection experiments in which we give the subject a brief tap to the finger and we ask them, did you feel it or did you not feel it? And this is data from an MEG experiment where we've applied a standard inverse solution technique to isolate the contribution to the signal from the hand area of S1. And this is the ongoing signal in S1 before we tap the finger. And I've applied here a common frequency analysis, and you can see that there's a high power oscillation in this 15 to 29 hertz band that we call a beta rhythm. And one of the main questions we were interested in is, does the power of this rhythm before we tap the finger impact the ability of the subject to feel that tap? And the answer to that question is yes. And so what I'm showing you over here on the right is we average the power in this pre-stimulus time window across the frequency band. We sort from low to high power, and we look at the detection rate as a percent change in hit rate from the mean. And what we see very clearly is that the higher the power, the less likely the subject is to feel that tap to the finger. And the real question of interest is why? What is it about the neural mechanisms that are creating this oscillation that lead to the subject saying, I didn't feel that tap to the finger? Now, before we can answer that question, we have to take a step back and understand our data a little bit better. I've mentioned to you that here we're averaging this signal, but on any tap to the finger, the brain isn't averaging, and there's this ongoing oscillation that we need to understand. And so what we found, taking a closer look at the unaveraged data, and here's just 10 examples of 1 second of this pre-stimulus period, what we see is these beta oscillations emerge transiently. They typically last less than 150 milliseconds. And it's only because of averaging this signal that we get this false sense that there's this continuous ongoing oscillation. Now, this transient nature of these macro scale oscillations appears to be a very robust phenomenon. So here I'm just showing you several examples from the literature where oscillations in different frequency bands appear to be transient in nature. And I don't want to go into detail of any of these studies, but just want to point out that it's been observed in motor cortex in this beta band. It's been observed in gamma rhythms in visual cortex and sensory motor cortex, alpha rhythms in sensory motor cortex, and this is a study looking at infants. And so these observations occurring at the macro scale outside of the head. And an important open question is this a real brain phenomena, or might it be due to an issue with our instrumentation and a signal to noise issue, where we just don't have the right signal to noise to see that, in fact, these are continuous oscillations, and we're recording them as these transient events. And the other important question is, what is the spatial scale over which this phenomena emerges? And there are some clear paths forward to answer this question. One is that we need to be developing improved imaging techniques that have better signal to noise. And one promising direction in this area is in optically pumped magnetometers, where we can get the sensors closer to the head, and presumably that will help improve our signal to noise. Now, along with developing improved imaging techniques, we have to also be developing the inverse methods to apply these techniques and the software that's necessary for the community to be able to use this technology in a meaningful way. We also need to be combining these human experiments with invasive recordings or other imaging modalities that can get at the underlying circuit activity. And in my lab, we've been focusing on combining these techniques with invasive recordings and other imaging modalities. And so I'm just going to give you an example of what we're seeing for these transient beta events. Here on the left, I'm showing you our human somatosensory data. And on the right, I'm showing you similar recordings from local field potentials in the somatosensory cortex of mice. And this is data collected in the lab of my colleague, Chris Moore at Brown. And we've been quantifying features of these beta events across these tasks and species. And what we find is that there's remarkable homology and things like the rate of these events in the pre-stimulus period, their amplitude, their duration, suggesting that's a very robust phenomena that's conserved across species and recording modalities. We've also taken a look at the underlying waveforms that are creating these high power beta events. And again, here we find a very robust phenomena that's conserved across species and recording modalities. So here I'm just showing you the overlay of the time domain activity from 50 of these high power beta events. And you can see that it has a very robust waveform shape that's dominated by this negative deflecting peak that lasts 50 milliseconds. It looks like an event, and that's why we're calling these beta events. Very similar findings when we look at the local field potential and somatosensory cortex of non-human primates, and this is data from Charlie Schroeder's lab at the Nathan Klein Institute. So these beta events have a stereotypical waveform shape that's conserved across the species. And so now we know that at least for beta, these rhythms do appear to be transient, and it's not a signal to noise issue with our measurements. But this leads to another question. What does this imply for how these transient oscillations influence behavior? For example, I've shown you that on average, this beta power is inhibitory to perception. But what does that mean in relation to these transient oscillations that are occurring at different points in time? And so to answer this question, one thing we have to do is we have to be developing new ways of thinking about these rhythms and quantifying how properties of these transient events relate to average differences in power or phase locking and how these correlate with behavior. And there's lots of ongoing efforts on this front from our lab and other labs. In the power domain, we've been developing methods to quantify things like the rate of the event, their amplitude, their duration. Bradley Wojtek's lab has been developing methods to quantify waveform features like their shape, whether they're sinusoidal, non-sinusoidal. In the realm of phase locking, Idan Tal and Moshe Abeles have been developing point process methods that are similar to the methods that we're using to look at the coordination of spiking activity between neurons. And Mark Woolrich and colleagues are developing some hidden Markov model methods to look at the coordination of transient bursts across area. So the field is now transitioning to a new way of thinking about brain rhythms. And next, I'm going to show you again an example of how we've been applying some of these methods to understand how these transient events relate to tactile perception. And so here, remember now that on average, if we have high power, we're less likely to feel that tap to the finger. But there are several features that could be contributing to a difference on average in low versus high power. It could be that on a low power trial, we have one event, and on a high power trial, we have more events of same amplitude, and this would average to high power. It could be that we have the same number of events, but the events are higher in amplitude, or their duration, or their frequency span is changing. Any of these could underlie a difference in low versus high power that we see and ultimately impact perception. And what we found is that in our data, a higher rate of the pre-stimulus beta events is underlying our high average power. And this is predictive of non-detection in our tactile detection experiment across recording modalities in species. And so now we have the follow-up question of why? What is going on in the underlying circuit to create these transient events? And how do those mechanisms potentially causally influence perception? So for this, we're going to turn to our modeling framework, and we're going to simulate the underlying neural mechanisms that can create these observed beta events. And the first thing to point out is that the primary electrical currents that generate MEG and EEG signals come from the intracellular current flow and these long and spatially aligned pyramidal neuron dendrites. And so our modeling framework contains generalizable features of neocortical circuits under thalamicortical and cortical drive. And in practice, what we do is we simulate trains of action to potentials that excite excitatory synapses in the local network to drive current flow up and down these dendrites. And so the first thing we need to understand is how does current flow up or down these dendrites relate to this waveform of the beta event? And we can get this information from our inverse solution methods. And when we apply a standard minimum norm inverse solution method, what we've discovered is that during this prominent negative deflection, current is flowing into the cortex or down the dendrites. And so now we can use that information to develop and test hypotheses on how the circuit activity creates that waveform. And the punchline of what we found is that the model is predicting is that the beta events emerge from the integration of excitatory synaptic input through these two pathways, such that the supragranular input is strong and it lasts 50 milliseconds. That pushes current flow down the dendrites for 50 milliseconds. Now, because this is a detailed model that includes microcircuit information, we can go in and test these model predictions with invasive recordings in animals. And indeed, we found support in both mice and monkeys that these model-derived predictions may be valid. We also now have some support from layer-resolved human MEG. Now, our model is further predicting that the reason that these transient beta events are inhibiting perception is that they're recruiting inhibition in the supragranular layers. And this inhibition is decreasing the ability for that sensory information to get to primary somatosensory cortex. And those are still predictions that need to be validated. So modeling is a really powerful tool to connect what we record outside of the head to the microcircuit activity. But there's also lots of challenges in developing and using this modeling framework. They're really computationally expensive, and we have to think about ways to get these types of modeling tools in the hands of the community, and we're trying really hard to do that with our HNN software tool. And so with that, I'm going to conclude and summarize some of the challenges and opportunities in decoding these human signals at the signal trial level. So I've shown you that complex analysis methods and averaging applied to interpret the relationship between these signals and behavior can create real challenges in connecting to the cell and circuit level activity. And one path forward is to be developing a better understanding of what's going on in a single area and to use that as a building block for understanding these more network complex phenomena. In non-average data, these prominent low-frequency oscillations often emerge as these transient events. And so to understand how this is relating to behavior, we need to develop improved methods to image the spatial and temporal structure of these events and new analysis methods to quantify event features and how they relate to changes in behavior. Lastly, I've shown you how biophysically realistic modeling can be a really powerful tool to uncover circuit mechanisms and their potential causal impact on behavior. But there's lots of challenges here. And some of the things we need to continue to do are to advance inverse methods that can help delineate the direction of the electrical currents, because that's what we map onto our biophysically detailed neural models. We need to continue to combine these modeling frameworks with other imaging modalities and invasive recordings, and we need to work to improve the accessibility of these complex modeling tools for the community who may not have experience in developing and using neural models. And so with that, I'd just like to thank my collaborators. I have an amazing lab at Brown, and everyone's contributed to this research in a meaningful way. HNN is being developed by a large team of researchers from Brown, Mass General, and Yale, and we're fortunate to have a network of collaborators that have enabled the interdisciplinary research that I've discussed today. So thank you for your time.